Hello everyone, welcome to Bioinsights Academy by Yasmin and Sarah. I'm Yasmin, and in this video, I'm celebrating the 2024 Nobel Prize in Physiology and Medicine awarded to the Victor Ambros and Gary Ruvkun for their groundbreaking discovery of microRNAs. Today, I walk you through the discovery, biogenesis, and function of these small yet powerful RNA molecules. If you're curious about this fascinating topic, let's dive right in. In the early 1980s, two postdoctoral fellows, Victor Ambros and Gary Ruvkun, were working independently in the Robert Horowitz's lab using a Hepatitis elegans, known as C. elegans, a tiny roundworm, as their mother organism. Their focus was on a gene called LIN4, known for regulating developmental timing, but the exact mechanism was unclear. LIN4 didn't code for a protein, which was unusual for a gene. This left the researchers puzzled. How could it control development without producing a protein? This mystery led to a groundbreaking discovery. Before talking about the discovery, biogenesis, and function of microRNA, it's better to talk about the C. elegans first and answer this question that why this nematode are widely used for genetic research. Well, actually, this C. elegans is a small, free-living nematode about 1 millimeter long that thrives in the temperate soils. Despite its small size, it has a specialized cell types like nerve and muscle cells similar to those in more complex animals. It's an ideal mother organism for genetic research because of its transparency, simple anatomy, and known genome. In fact, over 65% of C. elegans genes have human homologous, making it invaluable for studying gene function, including using tools like CRISPR-Cas9 and RNA interference. The ability to manipulate these genes quickly has made it a powerful system for investigating genetic pathways in development and disease. In 1993, Victor Ambros and his team made a groundbreaking discovery. They found that LIN4 didn't code for protein, but instead produced a small RNA molecule about 22 nucleotides long. This small RNA, later named microRNA, was the first of its kind. Ambros' team showed that LIN4 binds to specific regions of another gene, LIN14, preventing it from being translated into protein. This was a completely new form of gene regulation where LIN4 controlled development by blocking the production of proteins without degrading the mRNA. At the same time, Gary Ruvkun's lab confirmed Ambros' finding and further demonstrated that LIN4 binds to complementary sequences in the 3' UTR of LIN14 mRNA. This binding stopped LIN14 from producing its protein without destroying the mRNA itself, which revealed a novel form of gene regulation. Ruvkun's work solidified the role of small RNAs in gene regulation, showing how they could control a specific mRNA targets by blocking translation rather than degrading the mRNA. As you can see the figure at the left side of the slide, there are 3 worms which differ in their body shape. The diagram suggests that LIN4 and LIN14 genes are involved in a genetic regulatory pathway important for the proper development of C. elegans. Mutations in these genes cause a different abnormal physical forms in the worms, reflecting the disruption in normal development. In 2000, Gary Ruvkun made another significant discovery when his lab identified a second microRNA called LET7. This microRNA, like LIN4, regulated developmental timing, but it was highly conserved across species, from worms to humans. This discovery showed that microRNAs were not unique to C. elegans. They played a fundamental roles in gene regulation across a wide range of organisms, including humans. Thanks to the discoveries of Ambros and Ruvkun, we now understand how microRNAs function. MicroRNAs are initially transcribed as a long primary transcripts known as pre-microRNAs. Then, they are processed by the enzyme DROSA in the processor microRNAs known as pre-microRNAs. Then, pre-microRNAs are then exported to the cytoplasm where the enzyme dicer processes them into mature mRNAs. The mature mRNAs join the RNA-induced silencing complex known as RISC, which helps guide them to the target mRNAs. Once part of the RISC, microRNAs can either block mRNA translation or in some cases degrade the mRNA, influencing gene expression post-transcriptionally. Actually, microRNAs have been implicated in numerous biological processes. Some microRNAs act as tumor suppressors or oncogenes, making them key players in cancer development. Or abnormal microRNA expression can lead to developmental abnormalities in diseases. And also, altered microRNA activity has been linked to conditions like Alzheimer's and Parkinson's. The discoveries of Victor Ambros and Gary Ruvkun have revolutionized our understanding of gene regulation. By identifying microRNAs, they uncovered a new class of regulatory molecules that fine-tune gene expression. Their pioneering work has had an enormous impact on the fields like developmental biology and cancer research. These discoveries continue to shape how we approach gene regulation in both normal biology and disease. Thank you for watching. We hope you enjoyed learning about the discovery of microRNAs and their profound implications. Don't forget to like, share, and subscribe to Bioinsights Academy for more informative content on biotechnology. We'll see you in the next video.